was started, all because there is very little understanding of this technology and how it should be regulated, why it should be regulated. There are so many questions being raised on this technology, and we feel as Ugandans that it is very important that we have full understanding of what it is all about as we move forward to use it, so that we can use it in a safe, sustainable way, and in the interest of developing our country. It is very, very important that many people have very good understanding of what we are doing and why we are doing it. Why the scientists are saying what they are saying, why they continue with the technology, and why the debate is also on. The debate is global. The technology keeps moving forward. The debate also is going on. So we should realize the debate will never end, and actually things like OFAC will still be useful in this country even after, for example, GMO products are released and are on the market, so that people can still follow and they can voice what they want in terms of what is happening in the country. So once again, we have a privilege of listening to a seasoned scientist, somebody who has hands-on experience, somebody who has the training in the technology, and he's talking to us about a very pertinent topic, that is seed system. Many times the questions being raised are on seed. What about our seed? What about the indigenous seed costs? All those concerns are really center on seed. So I think the topic tonight is very pertinent, and we are looking at all of you as ambassadors. After you have listened to the presentation, to this presentation, and people who are pro-biotechnology, to talk to others about it, and also always to feel free to get more information from the scientists if you are not very sure. We are hearing in some corners people are saying things are not clear, they are getting confused. Don't allow yourself to be confused. The scientists are there, the regulators are there to give you the information you need. If there is an area which you are not clear about, kindly feel free to approach particularly narrow National Council of Science and Technology, because these are the government institutions responsible for the biotechnology and biosafety. They have all the full information. So I think it is very important that you have full understanding, because we are looking at all of you who have had an opportunity to listen to people like Andrew Chigundo to be ambassadors of this technology. So without taking up too much time, it's my pleasure to welcome you all to this lunchtime meeting and open the meeting and invite Dr. Andrew Chigundo to come and give his presentation. Thank you so much.
Thank you, Councillor Sande, for the introduction, and thank you, Teresa, for the opening remarks. Thank you, OFAP, UNCST, AATF, for inviting me to give yet another OFAP presentation. And my intention this afternoon is to present some basics of seed systems and where I feel the new technologies of based on genetic modification will fit into that system. Perhaps there's people in the room that understand seed systems much better than I do, and I would encourage those of you to listen in, correct me where I'm wrong, but also let us engage in a discussion so that we all start to figure out and place these technologies where they need to be. Having said that, the seed system in Uganda is evolving, and I think I will try also to highlight how it is changing from the old to the new, and how GMO technologies we are currently developing and others that might come will fit into that system. As Teresa just said, the discussion heats up a lot, especially as regards seeds. And my intention also is to try and create a picture, a background that allows us to appreciate the nature of seeds, the types of seeds, why certain seeds are the way they are, and that might also help us to understand how best we can discuss this subject. Before I start, I just want to let everybody appreciate that seed systems are complex, biotechnologies are complex, breeding and breeding systems are complex, and we should rather discuss on specifics rather than discuss broadly. You hear a discussion, oh, GMOs are like this, GMOs are like this, but if we would rather say the particular GMO you are referring to, then we can better understand the discussion. And as you will see, even in the seed system, it's much better appreciated when you discuss on a case-by-case basis. Say, if you want to discuss seed in cotton, or seed in maize, or seed in banana, you can appreciate that everything will be different, and then the discussion moves much better. I work for NARO at the National Agricultural Research Laboratories based at Kawanda. My original training, I started as a pure biologist. I did botany and zoology at Makerere, and then I did a master's in plant breeding. So I'm a plant breeder myself. I'm an innovator of seeds. So that's why I said people in the room might understand the further chain of seed systems better than I do. But these days we've continued our breeding using genetic modification, and I will show you what we're doing at NARO, and we'll discuss the issue. So my outline will first of all address what a seed is and how plants reproduce, some breeding and hybrid seed systems, and then I'll show you briefly the two main seed systems in Uganda. Then I'll also show you some GMO activities going on and where we see which particular ones might come in early. I'll also tell you what I think where in the system these GMOs will fit, some challenges in the seed sector, and I'll also present some way forward statements. So a seed is basically a reproductive mechanism of flowering plants. All plants desire to maintain or continue their species, and to do that they must find some way, some mechanism of reproducing themselves. A basic seed contains an embryo plant, and in that, together with that embryo, is usually a food source. Maybe an endosperm or a cotyledon that supports the embryo to establish. That is, if it is planted or if it finds a suitable medium for it to establish. But not all plants produce seeds. Some produce spores and others reproduce vegetatively. So even vegetative reproduction is a form of mechanism for plants to actually survive regeneration. It's the most important input in an agriculture system on which other inputs depend for improved agricultural productivity. So if you have bad seed, it doesn't matter how much fertilizer you will add to that bad seed, you will still not get the ultimate productivity. However much you irrigate bad seed, you still won't get the ultimate productivity. So it's really the first and most important input into an agricultural system. And plants reproduce mainly two ways. We say sexual reproduction and asexual reproduction. So under sexual reproduction, we have plants that cross-pollinate. I'll show you some detail of that, and other plants that self-pollinate. Under asexual reproduction, we have plants that basically use vegetative means to propagate, such as cuttings, suckers, and some plants normally on pieces of their tissues, somatic cells, to produce new plants, such as these plants. I'm sure you've seen this before. It's a Bryophyta, the leaf just drops on the ground, it pops new plants, even if it's not seed. So it's also a unique mechanism in this particular plant to ensure it survives generations. Some plants have sex. Those are the sexually reproducing plants. And they have plants to do that more or less like humans. And the leaf is the sexual organ of plants, but it also has usually both male and female organs or structures. We have anthers, which produce pollen, and these are the male parts of the plant. The pollen divides into cells, individual generative cells, and as that division takes place, each pollen has half of the genetic material of the plant. And the pollen goes on to fertilize the ovum through stamen, stigma, and the ovaries are produced in the ovules are produced in the ovary, and those ovules become seed, and that seed can germinate back to another plant. On the other side is the female structure. They have an ovary. The ovary also produces an ovule, and as the ovule sits inside the ovary, the pollen will germinate and fertilize it. So the ovule and the pollen cells each have half of the genetic constitution or the genomic constitution of the plant, which means at this stage in sexually reproducing plants, there is a mixture. The half of the genetic constitution of the male plant is mixed or is added to half in the female, and usually they mix the genetic background of the male and the female. Typical of animals, and I'm sure it's much more appreciated in humans. Other plants use asexual reproduction, and we know a few of these. Some have runners, such as in strawberry, and as the runner goes along the ground, new plants form. Potatoes, right? From these tubers that's produced. So the tubers have buds on them, and each of those buds can germinate into a plant. The most common ones, food source in Uganda, are bananas, and as we all know, they produce by suckers, such as these ones, and cassava, which is planted by using cutting. So you just cut the stem has buds, and each of these buds can germinate or can produce a whole plant. For us who are used to breeding, we have the rare opportunity of seeing the seeds of these two plants. Some wild bananas will produce seeds, such as there, and also some wild cassava. If you need to a conventional breeding, you can find some wild varieties that produce seed, and that's how they look like. So this mechanism of reproduction is common in banana, cassava, potato, and sweet potato as some of our main food crops. So back to pollination and sexual reproduction, which is usually not properly understood. And I took this slide to give us an understanding of the processes. First of all, we have pollinating agents, wind. So wind blows around the pollen from one plant to another. We also have insects. And as insects visit different plants to feed on juices that are produced in the plant, they also carry pollen around. And the different types of pollination, said earlier, there is self-pollination, which means this particular plant has to pollinate itself, which means pollen from the male part to the female part of the same plant. Sometimes it is the same flower or different flowers but of the same plant. And then there are plants whose main mechanism of reproduction is cross-pollination, which means that there are mechanisms within the structures of this plant that ensure that successful pollen or successful seed can only be produced if pollen crosses from one plant to another. So, and that is common in maize, in coffee, some people don't know, in sunflower and sugar. These are most of the important crops we have that use mostly this method of seed production. Now, humans have also appreciated that sometimes there are barriers to pollination, and also for their need to pollinate or to breed plants, they can artificially assist plants to pollinate, either for the purposes of breeding, but also we know some crops such as vanilla that will never produce fruit unless it is artificially pollinated. Down here is a list of the most common self-pollinated crops: common bean, tomato, groundnut, rice, soybean, and millet. But which means that because these plants are self-pollinated, they do not essentially differ in their genetic constitution. You remember when I told you about cross-pollination? It's half of the genome from the male plant and half of the genome from the female plant. But in the case of plants that self-pollinate, even if they have this halving of the genomes, but remember they are pollinating themselves, it means that basically the genomic constitution remains the same. Okay, if need be, I may come back to that slide later. But breeders, as I told you earlier, have found a way in which to force these self-pollinating plants to pollinate for the purpose of breeding. And there are a few mechanisms. So we can do selfing. Again, this is a brush. You can get pollen from the anthers and make sure it goes onto the stigma. And also you can simply remove anthers so that the only pollen that goes onto this plant is coming from another plant, which means you intend to cross these plants and kind of change change their genomic constitution so that you arrive at some new varieties. And this gentleman, who is a priest and lived between 1822 and 1884, his name Gregor Mendel, is the father of this genetics. He's the gentleman who first described genetics and the transfer of traits of genetic material and heritable traits by cross-pollinating some pea plants that he was studying in a monastery in Austria. And since then, we've been breeding crops using this method of cross-breeding to try and get their gametes to fertilize different plants and therefore combine and look for benefits in the new recombined genomics or the new recombined genes, looking basically for traits and characteristics that will improve the plant for resistance to diseases, to pests, and probably high yield. That's why there was an evolution of a hybrid system. And I attempt here to explain hybrid seed system. Hybrid seed is the seed that is produced by cross-pollinating, controlled cross-pollinating. It began with maize. Maize was the first and still is the biggest and most important hybrid seed crop in 1920, when it was discovered that if you have two plants that are pure in their genetic background, and if you cross them, such as in this process, they combine the genetic benefits into the seed that will be produced. Because when they grow the seed that came out of that cross-pollination, the plants looked bigger, were more vigorous, the yield was higher, and the plants tended to also be very uniform. So that was the discovery of a phenomenon called hybrid vigor. When you combine the genetics, when you combine the genetic background of two pure lines, those genetic benefits combine in the seed that is produced after that cross. So the system evolved that when you do controlled crossing of two pure lines, you will produce seeds that will germinate into a wonderful, vigorous plant. And because of the farmers could not do controlled crossing themselves, they would not have access to pure lines, there was an evolution of a commercial seed system. Because the seed company would know how to do this, would know how to do the controlled process, and would sell the high yielding seed to the farmers and make some money. So around that time, in the 30s, that's when hybrid system started, and actually that was when commercial seed companies evolved, mainly because of this hybrid seed system. Farmers cannot do the crosses themselves, and the company and competent breeders who were employed by these companies could produce hybrid seed. So that seed is then planted to produce a crop that is harvested for use. It's vigorous, it's an F1, vigorous, high yielding, and the farmer would have increased yield by planting that hybrid seed. However, if seed from that F1, if seed from that hybrid plant is planted, as I told you, there's cross-pollinating in especially maize. That cross-pollinating will have and will reduce the vigor that was seen in the seed. So we recommend farmers not to replant hybrid seed because they will see a drastic reduction in the vigor, because that cross-pollinating, that sexual reproduction has halved the genetic constitution of the parent and of the parent plants. So in a field of F1 seed garden, there's cross-pollination by wind, by insects, and that sort of dilutes out the vigor. So we recommend farmers not to plant seed from a hybrid crop. The vigor and the high yield and uniformity that these seeds produce was actually a major factor in the green revolution of the last half of the 20th century, especially in Asia, on maize, on rice, and vegetable crops. This is another illustration of the same thing. Ordinarily, in maize, it has a male flower up there and a female flower down here that becomes the cob that we use. Pollen preferably moves from the male flower to the female flower of another plant. And if this cross is done in a controlled fashion of one inbred parent and another inbred parent or pure parents, you will get seed, and when that seed is planted, the plants are more vigorous than the parents, and the yield is much higher than even the parents, because that seed has combined the genetics of these two. However, if you take seed from this, because of random sex and cross-pollination in this field, you will see a drastic breakdown in the yield benefit, and also quite an ununiform yield, as you get back some of the characteristics of the parents. So because of that, there's a misconception that this seed doesn't germinate. That's not true. It germinates, but there will be a drastic reduction in yield. This is a picture which illustrates parental material. These are inbred lines, they are pure lines. You can obviously see they are very different. One is much bigger than the other, but when they are cross-pollinated, the seed that comes from these will be more vigorous. Will be somewhere here, higher than both of them. And these are some of the processes that are used to control crossing. You can do by bagging, so you can bag the male, what you want to be the male parent, so that pollen only goes to the other plant. So we use bagging to control crossing, or we can use detasseling. And in this field, you can see some lines still have the tassels, the male flowers, and other lines don't. They have developed tractors, special tractors that can go through the field and cut off all the flowers of what lines will be female. So that pollen in this field is coming only. And this whole field, the seed that comes from this whole field will be hybrid seed. So, some seed system in Uganda, there's two seed systems. There's a formal seed system and an informal seed system. Now, the formal seed system basically starts with the government, and then government has a regulatory role of the research and developers, the breeders, NARO, and the universities. It also has a regulatory role of the private sector. The private sector mostly constitutes the seed companies, who sometimes work with research, either by sharing the experiments, or they obtain breeder seed from the seed company, from the research. And then seed flows to the extension system, which is also a government system, either from the research, the developers, the breeders, or from the private sector. And then that eventually flows to the farmer. The farmer is bombarded by seed from extension. These days, research and universities are required to engage with the farmers, so they're also bombarded, and also the private sector bombards them with seed. This formal system started way back in 1960, when the government and its development partners realized that the farming community in Uganda needed to benefit from the yield advantage of hybrid system or hybrid seed. But because there's commercial interest in this system, there's need for regulation. So the government had to develop a regulatory system for this seed, and it's currently being regulated by an agency called the National Seed Certification Service. Then around 1990, the seed sector was liberalized, which means that the government preferred that all seeds should be produced by private sector. It had some advantages and disadvantages, as we will see later. But it also has issues of intellectual property, because breeders and companies that breed hold the information of the breeds because they want to be in the competition, to beat the competition. They want to be ahead of the competition. So there is secrecy, there is protection of intellectual property. And as we said earlier, this system is mainly concerned with crops that are cross-pollinated, from which hybrid vigor can be achieved. And again, examples include maize, sorghum, and several vegetables. The other system is the informal system, fairly small, farmer to farmer, exchange of seeds. Farmer sells seeds, so the same farmer can keep selling his seeds, can share or minimally sell, there's a little bit of sell to another farmer. And right now, there are a lot of NGOs or agencies that are involved in this informal seed sector. Important things to note is that it constitutes 80% of seed planted in Uganda right now. I think our neighbors in Kenya are much lower than this. They have captured, or they have adopted a lot of the hybrid technology in Kenya. And even in Uganda, because of our eastern brothers and sisters being closer to Kenya, we find hybrid seed much more used in eastern Uganda up to around the Busoga sub-region, much higher than further this way. But we think that it's moving towards the west, and many more farmers, the west side of Kampala, are increasingly using hybrid seed. So, yes, there are some farmer organizations that want to organize themselves in producing seed, but again, because they are not companies, they are not regulated. It's mostly characterized by self-pollinated crops such as millets and legumes, and of course vegetative crops like cassava, banana, and potatoes. There are again seed schemes that are coming up at community level, and small businesses that few people, youth, are starting here and there to try and commercially sell seeds. I'll show you later about one of the activities we are doing at Kawanda. We are developing a community seed system. Again, it's based on these self-pollinated legumes and millets where farmers can set up, or we help farmers to set up a community seed bank, and they can sell their seed there. But as I told you, this system can only be for non-cross-pollinating seed. Otherwise, cross-pollinating crops, it's difficult to maintain the genetics because every time you plant, the genetics is just mixing up and reducing on the vigor of that seed. So that brings me to our efforts in biotechnology and genetic modification. The genesis of NARO getting involved or using this technology for breeding comes from the fact that most of our crops are vegetatively propagated. If you see the yield potential of food or the amount of food being consumed, it's mainly bananas, cassava, potatoes. These are the big ones. And they suffer a lot of biotic and abiotic constraints, diseases, pests, drought. The input is low, the varieties are few, and these varieties have been worked and worked and worked by that continuous replanting. So we realize that resistant varieties are very important in this sector or this type of crops. And NARO has been using crossbreeding in the past. And as I showed you, we look for wild relatives of these crops. The pictures here, you see banana which is seeded, very miserable fruit, and we try to cross it with some consumer preferred varieties, and looking to introduce yield benefit, looking to introduce resistance to diseases and pests. However, what happens with this system is that because of the complex genetics and because of working with wild parents, there is a lot of undesired characteristics that come along with this type of breeding. And many of you already know about some of the banana hybrids that were released that farmers really don't like very much. They are high yielding, but they really don't taste like the food people would like to eat. And those are the challenges because we have worked with wild types to introduce resistance, but there are undesirable characteristics that come along. So that forced NARO to start moving away from conventional breeding, especially in these crops, and look for other more precise breeding methods. And certainly genetic engineering was the one. It offers leverage over these breeding constraints, timely delivery of farmer-preferred varieties, and also we can now introduce some of the traits or some of the characteristics that we couldn't do with conventional means, such as improving the nutrient content, vitamins and minerals in these crops. So basically, this slide shouldn't scare you very much. I'll go through it briefly. So basically, you have a gene. You identify the gene for resistance, and there's two main methods of introducing the gene. Using agrobacterium, which is a bacterium that naturally transfers genes into plants, or a gene gun. But we don't use the gene gun anymore. Although what normally happens in the gene gun is that you have DNA or the genes coated on some gold, micro gold particles, and then in a chamber there is something like a gun actually that shoots these gold particles into the cells. And as the cells multiply, they form a plant that is transgenic. But our preferred method these days is the natural one, which uses agrobacterium, a soil agrobacterium or soil bacterium that normally lives with plants, and found that it introduces its own genes. So we use it as a vehicle or as a tool, a vector, to transfer the genes that confer benefit to the plant. And the bacterium infects the cell, it transfers the genes to the cells, and then we can regenerate those cells into a normal transgenic plant. We've done that for mainly bananas now, cassava, and several other crops are beginning to use this technique to develop resistance and higher performance. These are some pictures of how we do it in bananas. Again, you'll see that we introduce this agrobacterium, which has the gene of interest, onto banana cells. And you see banana cells which survive are collected and germinated back into normal plants. And these small plants in the test tube are transgenic. We later can grow these plants up into normal plants and test them in a field experiment. These are some other pictures of how we do it in cassava. Again, we have some cells on the petri dish infected with agrobacterium that has the gene of interest, and those plants can be germinated back into normal plants. And we generate a lot of these plants and again test them in the field to see if they are becoming resistant. And so these are some of the field experiments that are currently being undertaken or have been undertaken in Uganda. In Serere, which is actually now a district, I think, we previously tested cotton that is resistant to cotton bollworm. We also tested cotton that is resistant to herbicides, which means farmers can spray herbicide to kill weed and the cotton doesn't die. It's a wonderful technology and farmers are really excited about it. Right now in the world, about 80% of GMO crops being grown are herbicide resistant. Because the farmer just has two hands. If a typical farmer would ever be able to increase acreage without tractors and that type of thing, herbicide tolerance is one of the technologies that will help the farmer be able to increase the acreage. In Namlonge, we tested cassava with resistance to brown streak virus disease, cassava with resistance to cassava mosaic disease, bananas at Kawanda, tested bananas with black cigartoka disease resistance, bananas with banana bacterial wilt. This one is my favorite. Just last month we published this work in Nature. Nature Biotechnology is the highest ranked scientific journal in the world, and we were privileged to have published our work there successfully. We have seen 100% resistance at bacterial wilt in bananas, and not just normal bananas, but our matoke bananas. And for some of these technologies, we're just waiting for the law to allow us progress to farm. Again, at Kawanda, we tested biofortified vitamin A and iron bananas, and other bananas with resistance to nematodes and weevils. In Kasese, we've tested cotton again, the same cotton we tested in Serere, but a very interesting project going on in Kasese is maize with resistance to drought, to water stress. That one has also offered almost 40–45% yield benefit, even when there is less moisture, which means that farmers should still be able to make a yield benefit from this seed or from these varieties, even when the rains are short. Many people are concerned about with genetically modified crops and organisms. This table just highlights some of the tests, some of the risk assessments that all these genetically modified crops have to go through, and all these are specified in the law that has been proposed. They fall in 4 basic steps. For the parent crop, that is the crop that you are going to genetically modify, they have to identify the genomic performance of that crop, the geographical distribution, where does it normally grow, the history of safe use, which means that if people have already been eating this food, and the composition analysis of the parent crop, so that we can see how it has changed. Then the next stage, when you are doing the transformation, we have to look at the description of the donor, which means the plant or the organism from which the genes were extracted. If it is the same plant, then the level of risk is much less. If it is from an animal to a plant, then the level of risk might be different. And then a description of the vector or the system you're using to introduce the DNA. We need a transgene delivery process. How have you transferred the genes? Characterization of the DNA that you've used. Insertion site, where the DNA or where the gene that you introduced has been integrated. And then in the third step, we have to look at the product. What is the structure, the identity, characterize the product of that gene, the mode of action, the toxicity, the allergenicity of the product of that gene. Then once the plant has been developed, transgenic crop, it has to be assessed for safety. We identify it phenotypically, how does it grow, is it normal, what are the agronomic benefits that you've introduced versus those that were already there. The composition analysis again, and how different is it from what it was before you started. And the safety analysis goes on, and in some cases, there's need for animal studies. So feed it to the animals and see the animals don't die. So this is the brief of the safety analysis that must go before these crops are deregulated or provided for commercial use. And I must say that for the first time in our civilization, this process has to be done for crop varieties, which means, in fact, it has already been shown by some agencies around the world that because of this rigorous evaluation of these crops, they are already safer than even some of the crops we're already consuming. So where does all this GM technology fit in the seed system that I earlier described? For us at NARO, we follow a stewardship principle. We want to steward our technologies throughout the product life of the plant or the crop or the variety. We steward, or we use stewardship principles and gene discovery, so we are cautious about where the genes come from. For example, our preferred source of genes is the same crop. If there are some resistant individuals, preferably we source the gene from there, or from crops that people are already consuming. The process of developing the plant, the seed production, so we're also cautious about how will the seed be produced to be distributed. And as I showed you earlier in the former seed system, at this point, we look for partnership with the private sector, because also, as I showed you, I prefer or that the seed system is driven by the private sector. So that then goes to seed marketing and distribution, crop production, crop utilization. All these stages we are with the farmers to see what the farmers are complaining about, to see what the farmers are excited about, including utilization and product discontinuation. So at the last stage of this life cycle, farmers don't like it, we can discontinue. Or if in the lab we have new versions which are better, and I think we will start to combine some of these traits, we can go back to the farmer with a different seed and say, okay, the one that you have been growing is not as good, we bring you a better one. So, in informal seed sector, concerning self-pollinated and vegetatively propagated crops, or the formal sector. As we say, the formal sector has mostly private companies. But as you can see, all these crops are mostly in the informal seed sector, which means that they do not attract a lot of monetary benefits and companies being interested in trading in this seed. And farmers, either as organized entities or farmer groups, can continue to work with this seed without intellectual property intervention by companies. So the challenges in the seed system, someone told you that the seed sector in Uganda is evolving. Because of these challenges, we see that the government is working on a new seed policy, and after the new seed policy there will be a revision of the Seed Act, and there are a few new legislations that are coming up. There are challenges of enforcing the regulations that currently exist. There is inadequate research and development of seeds, which means lack of intellectual property protection. Many people are not excited about producing seed which people will just take, produce, and there's no monetary benefit. As a result of structural adjustment in the past, the government preferred that the sector is led by the private entities, but this has created a bit of a problem because they do not want to engage in those self-pollinating seeds because the farmer buys once, he goes, sells seed, and it doesn't come back. So the companies do not like to sell beans, they do not like to sell rice, because these crops farmers can save seed. So those crops have had a problem of getting enough seed in the system. And now we think maybe the private sector or the public sector must get back into the seed business and start to promote the seeds of those self-pollinated crops. Tissue culture has come around, it's growing slowly, but there's still a concern of cost of tissue culture plant rates. But we think as more and more people engage in this technology, the prices will go down. There's general lack of awareness. People really don't appreciate seed systems, and I think for some of us who are involved in this sector, we need to increase the awareness. And also the informal seed systems are not recognized and therefore not regulated. That's also important because as it grows, farmers will start to also sell and distribute material that is not properly regulated and is of poor quality. So we think that if there is a policy in place, and as I speak now, that policy is being debated, it's probably going to be approved soon. We need to increase capacity for monitoring and quality control, certification, and seed tracking. I know that the ministry is battling with this, and they are looking for ways in which to improve this. One of the ways is perhaps to create a mobile application that the farmer can SMS the code on a seed bag, and the SMS can respond with a confirmation that this is good seed. Something like that. Otherwise, it's difficult to have a policeman everywhere, and maybe also to certify the traders themselves to see how to improve on quality. As I said, I think public institutions need to increase their efforts in seed production so that they can address the other self-pollinated seed for which companies do not have much interest. So NARO is already beginning to do that, and I know Mackay University is the home of soybean. Up to now, they're probably the only ones that are producing and selling soybean seed. There's need to improve the formal sector, the formal seed sector, and the policy that has been drafted now recognizes a new type of seed that was proposed by the FAO some time back, and it's called a quality declared seed, which means that that informal sector needs to deformalize a little bit and start to characterize the seed that they produce as quality declared seed. It also has to be monitored and certified, but much more at a lower level, so that when farmers start to produce seed, they can also be recognized that their seed is of good quality. Increasing public awareness. And one of the things I find people do not appreciate about seed is that seed is a living thing. I have seen companies and people who hope to be distributing seed, just carrying seed on an open truck from Kasese to Kampala. But by the time that truck has driven that whole distance, and it's very hot and the sun is shining on these bags, you probably already have reduced 30 to 40% of the germination potential of that seed, because the hot sun over 2 days of movement has actually killed the seeds. So people don't appreciate that seeds are living. They have to be carried properly, stored properly, so that they germinate at the end when farmers are distributing the seed. We're also happy about the new plant variety protection bill. or law that was passed early or late last year. We think that it will help stimulate seed innovation, so many more people, some of the young people we teach breeding and genetics, they look and they can't find what they can do with themselves. So we're already trying to show them that they can actually start small seed companies and innovate, create some innovative seed that can benefit farmers in different ways, and then they can employ themselves that way. Also, it recognizes for the first time, and I think Uganda is well known for making good laws. For the first time in many countries around the world that have this law, do not recognize native seed, do not recognize that farmers can also protect the seed that they have grown over generations. So for the first time, I've seen this law recognizes and has a category of farmer seed, and those farmer breeds and seeds can actually be protected under that law. And any proceeds that arise from any commercial benefit from community-owned seed should actually attract money, and that money can go to help those communities. So my last slide is again about our effort to increase community seed banking. And this is an example of one community seed bank that we inaugurated in Nakaseke just early this year. So farmers basically bring their valued seed to this community seed bank, and it is sorted, it's graded, and it is stored. Any other farmers who want to use it are allowed to take some of that seed with an agreement to return 3 times the amount of seed that they have taken, so that we keep increasing the amount of seed stored there. But we also need to engage in awareness. So every now and then we have these seed diversity fairs and bring farmers around. They share knowledge about the seed, they share knowledge about how to maintain, how to grow it, how to register as members of the seed bank, and also participate in benefiting from the seed bank. So, thank you. That's all I had. And I hope Sunday we have some time for a discussion. Thank you.
Thank you, Andrew. That's been very nice. You've heard from him. He's a seasoned scientist in this area of seed. He has given his background. And I'm sure we love to learn, since this is often the reason why we come to such forum, to share, to get such knowledge that is science-based. Okay? Often you realize that our thinking at times is prejudiced by views that are not scientifically founded. So when we get such opportunity, unless they are not challenged, we should always embrace them. He has been very open as to the works of Naro that have been published in the journal. It's Nature. Exactly. I think that's a reputable journal, and it has not been challenged. It's well read, and when you rub shoulders or you move all over, you find people will tell you, those who read will always refer to Uganda as a success story in that area. So please, we have come to a very interesting moment, I guess now, where we are going to freely engage in on aspects or areas where we want to learn more or where we want to know more. We shall be keeping time. So there's one ritual that we did perform. That was introduction, self-introduction, because time was not our best ally. So when you raise up your hand, you want to say something, please introduce yourself as well. Thank you.
Thank you, Chachi Kundu, for your wonderful presentation. My name is Nassim Mugwanya from Uganda Biosciences Information Center. I'm just wondering how the biotech seed is going to not fit within the counterfeit system, which is already a big problem with the current situation. Thank you.
No, that's good, thank you. Any other? Now, this is going to be a challenge. Someone has to talk, everybody has to talk, so as you are known. Introduce yourself.
Thank you, Andrew. I'm Kodimonge Abubeka. I'm also a research officer. This topic actually is very good to me because, as you know, farmers have a lot of concerns about this technology, biotechnology. So I don't know what are the efforts of OFAP and then other stakeholders in reaching out to the farmers, because you know those myths of is it terminator seed. So what are the efforts of OFAP and other stakeholders on reaching out to farmers, especially on biotech, agricultural biotechnology, and then the seed system?
Introduce yourself better.
I'm called Mwonge. I'm a research officer working with the serious program. Now.
Okay. Oh, before I take lead.
Okay. Thank you very much. I'm Josephine Namaganda from the National Agricultural Research Laboratories, Rwanda. Mine is not a question, but a comment. Andrew, thank you very much. This presentation is very timely. I noticed there's a very serious problem, you know, out there in the communities. It is very difficult for, you know, the general public to tell the difference between a type of seed and, you know, the genetically modified materials. For example, we always get problems when we want to give out tissue culture plantlets to farmers, and they always, you know, think it is genetically modified. So as we are sensitizing people, we should make it, you know, emphasize the fact that it is a type of seed, but it is not genetically modified. And those of you who are listening to the news NTV last night, a comment was made about the type of seed that was planted. I think it was at a farm in Mbarara. The farmer said he plants the long type of banana suckers. We normally, you know, advise farmers to use the pomes. First of all, they are easier to transport, and secondly, establishment is much better. But you know, as Sudil Biarhanga was making a comment, he said that the genetically transformed bananas are buried in the soil. So we should always make sure that such confusion is got rid of. Thanks.
You watch news, I also did that. Thank you. Yes, the floor is yours.
Okay. Thank you. I'm Emmanuel Mataunga. I'm the Executive Director of the Uganda Export Promotion Board. Two issues that I want to raise. And maybe I start from where my sister has left. Currently, we are having a problem of our exporters, particularly of horticultural products getting into the European Union market. And the tests that were carried out in the EU laboratories found certain organisms which are not supposed to be in the food. Now, when the exporters have come back to us, they are blaming the seed. And you are the seed breeder. So I hope that we may, as we do the science, you might want to start thinking of how maybe we dispel such perceptions from the public, or if indeed science is needed to address it, you start thinking of how to do that. Two, you have talked of the crops where you are doing the testing. You talked of cassava, cotton, banana, and maize. I was wondering, what's the criteria that you use when you are determining which crops to focus on? Is there usually some bit of commercial interest that is taken into consideration, or is it purely from the science perspective? Thank you.
My name is Geoffrey Ekongot. I am a digital media consultant. My question is, the genetically modified seeds, who owns the intellectual property? What are the licensing procedures for, say, the private sector to access this intellectual property? And then question 2 is, you mentioned the tests that you run. Do you carry out long-term tests? And what is your idea of long-term, so that we know the impact? Thank you.
Yes, there are very many, it's good, but please, other experts are also here. Take note of some of those questions so that you can blend. Yes, Edward?
My name is Edward Junirwe from Uganda National Council for Science and Technology. I would like to thank Dr. Chigudu for a very good presentation. My question is, some of the concerns of the GM seed, some people have raised, as you clarified, people are having concerns on whether the so-called indigenous seeds will still be available to our farmers once the GM seeds are introduced. But my question is, do we have the so-called indigenous seeds? Because it looks like most of the plants that we have currently, most of the crops that we have currently are not indigenous. So what are we protecting?
One other, then maybe the experts.
Okay, thank you. My name is Harriet Tian from the Ministry of Justice and Constitutional Affairs. My question is there such a thing as counterfeit seeds? Because farmers have complained that sometimes they're given seeds and they don't germinate. And if that doesn't exist, what is actually the problem with those seeds that don't germinate?
Andrew? Others can feel free. Yes.
Thank you. Thank you for those very good questions. I will attempt, and anybody who feels they need to supplement are more than welcome. So how will the GM seed avoid counterfeiting or a counterfeit thing? And that might also go to Harriet's question at the same time. I think, again, it will depend on the seed type, on the seed system it falls in. If it is within the informal seed system, I think that system is fairly not that much counterfeited, or you don't get a lot of fake, which is what I think you mean by counterfeit seed system. Because farmers are just keeping the seed to themselves, they do a little bit of sale here and there. And there's not so much commercial interest in that system. And again, the crops associated in that system are those that are self-pollinating, which means they maintain their genetic potential for a long time. Others are vegetatively propagated. Otherwise, I think for the other ones, the formal sector, I think they would still suffer the same problems we are currently seeing, because some traders just pick up whatever they can find and they put color and they pack. These days, the technology of printing packets and printing boxes is advanced. The original box looks exactly the same as what they have printed on NASA Road. So unfortunately, we still have to find cleverer ways of avoiding those type of things. For Harriet, I think counterfeiting is not the correct term. It's just unscrupulous traders and who just package anything. What would have been produce for eating, they just package it as seed and they sell it. On top of that are again the awareness that I spoke about. Even sometimes when this seed reaches even when this seed has come from genuine sources, it is not properly handled. If it is delivered, say, to a sub-county office, you find they have just put it in a garage there, they have not protected it, they have not put it in an aerated room. They forget that seed is a living organism. It needs to breathe, it needs a certain amount of temperature. If it is too wet, it will die. Sorry, if it is too wet, it will germinate in the buds. If it is too hot, it will die. Then if you distribute this seed later, representatives, if possible, even pay for them to come from up-country residences to come and engage in these discussions, especially the leaders. At least for them, if they get to understand what we talk about here, they can use the different methods to transfer this information further down. But as Teresa said in her opening remarks, the debate, in my opinion, will never end. And the example I have is that the US has been producing and eating GMO foods for the last 30 years, but even there the debate has not stopped. So we are not looking forward to the debate to end now, but I think that we can always continue. If we have the bill, the law that puts in place the regulatory mechanism, we can proceed with the technology. Yes, Josephine, it's true, it's very difficult for farmers to tell the difference. In my personal opinion, I think it would even be good if we, as we develop these seeds and this technology, if we could find a way of labeling, maybe with color. And the example I have, some of the bananas we have that have vitamin A, some of them, the leaves look orange, such as, because vitamin A, beta-carotene has orange color. And I think that is a good marker, so that people out there can say, oh, if you see that orange, it is the one with vitamin A. Then it even reaches the intended use much better, and people can tell the difference. But right now, whether it is GMO or not, it will be very difficult to tell the difference just from looking at the seed. Perhaps as the law comes in, and the NBC and those who will be regulating the technology might think of a way in which perhaps we package the seed and market it in a way that people can tell the difference. Emmanuel from Uganda Export Promotions Board, thank you for coming. It's, I think, the first time we are having representation from that agency. It's true, a lot of our produce that reaches the EU market and it is rejected. It is also true that the rejection is based on presence of contaminating organisms in the produce they are exporting. Sometimes it's toxins, such as aflatoxin. That's why our maize cannot even reach, some of the neighboring countries, because the levels sometimes are too high. But it's also not true that this is because of seed. I think that the traders who already know that they have to meet those stringent standards, they hope sometimes that somebody may not see, but then when they come and they are claiming to be compensated or issues like that, they say, oh, I know it was the seed. But it's not true, it's all from probably, again, the informal farming system. They collect seed from they collect their produce from different places. The cleaning process is not clear or not straightforward. And indeed, a lot of our products meet challenges to meet EU standards. Yeah. So one of the things I always think about is that if these traders would like to engage in increasing their export to this EU market, they perhaps need to start at the farm level. They need to work with the contract farmers. And as they work with contract farmers, they monitor a bit of the practices on farms. because you can collect tons and tons of your produce. If you go to this one farmer that has, a small organism that is, then you mix everything together, the whole lot will lose, will lose the standard level, and it will be a big loss to the farmers, to the exporters. What criteria do we use to determine which crop to develop, and is this scientific or just or commercial? For us at NARO, this is purely scientific. It is based on needs analysis. We try as much as possible to make sure that we go GM where it offers a clear benefit, especially in trying to solve the issues, the challenges that the farmers face on farm. Diseases, pests, and drought. However, some of the commercially produced technologies do offer a significant benefit in our opinion. As I told you, the herbicide-tolerant seed that we tested for cotton, we could clearly see a benefit to the farmer. And we partner with the companies to evaluate this material in Uganda, and hopefully the farmers who are like in the cotton subsector could benefit from this technology. So there's more of a scientific criteria than a commercial criteria, but we can't run away from the commercial criteria, because in any case, if we have cotton farmers, their interest is purely commercial, because we don't eat cotton. Was it Godfrey or Geoffrey from Digital Media? Who owns the intellectual property? This also varies a lot. For the stuff that we've developed here, we own most of the intellectual property, if not all of it. And we do a due diligence to make sure that the processes that we use, the methods that we use are open, are free of intellectual property. Unless it is so critical that there will be some intellectual property. So what we then do, we negotiate for access to that intellectual property. And then we co-own it with whoever has licensed it to us. But when the product will be available to the farmer, as a national research institution, we will provide it free of that intellectual property. And that's the basis of the negotiations, that this, we co-own the intellectual property, but for subsistence farming or for small-scale farming, the technology will be available in the seed, free of intellectual property cost, but it might be seed cost, but not for the technology. And as I said, this varies a lot. We also know, as I discussed earlier, that there will be commercial companies coming in to bring technologies for those farmers that need it and can afford it. And then that seed will certainly have a markup on intellectual property. And it will be for the decision of the farmer whether they want to afford that seed, or if the added cost of that seed plowed into their farming enterprise gives them a higher cost benefit. Or do we carry out long-term tests? Now, the decision to do long-term tests, again, is dependent on the regulatory agency. I'll give you an example. As I showed you in the table, the risk factors vary on case-by-case basis. So if for example, I took the gene from the same banana that we are already consuming, which means there's a history of safe use, then the requirement for the different safety tests is not the same as if the gene came from, say, another species. You see? So even the requirement for long-term tests would depend on what what technology, what product you are seeking to deregulate. Yeah. Yeah. Some cases, indigenous seed is worked out. Yes, indigenous seed. Indeed, the term is sometimes misused. Scientifically, we know that the only indigenous crops in Uganda are millets So we are, this part of the world is the origin of some of the millets grown and coffee. We also share part of the area in which coffee evolved, which means some of the Robusta coffees originated in Uganda. And in the world area or in the world system of conservation of indigenous seed, Uganda contributes only millet and coffee. So all the other crops, all the other food crops that we grow here have been brought from other parts of the world. So, but indeed, we eventually start to classify them as land races. So if a community introduces a seed, maybe way back, grandparents introduced this seed from somewhere, but they have grown it for a considerable period of time and just become part of their culture. We classify that as a land race, and we still conserve that land race in our various collections, the biggest of which is at Naro, at the National Seed Bank in Entebbe, where we have about 3,700 accessions. And I think that was about all. The other one for Harriet, I think I answered it in the first one. Thank you.
Yes. Thank you very much. Yes. What featured prominently in his answers was about the regulatory system, regulation. Why he hints on that is that this technology is regulated. There is a regulatory system. World over, under the protocol, that's the requirement that the technology has to be regulated. Thank you. The regulation has to be regulated. Sorry, the technology has to be regulated. This country has also that system in place. We have a regulatory framework. And to launch us to this, in our midst we have the chairman of the National Biosafety Committee, who has also joined us in this discussion. And I think, Prof, we are honored to receive you. This discussion has been very interesting. After following the presentation of Dr. Andrew Chigundu on seeds. We've missed the slides, but it's been very widely capturing indigenous, all the seeds and the developed seeds that he has been answering questions over. At this juncture, before I conclude, I'm going to invite you, more or less as a custodian in these things, and the chairman of the regulatory system, the NBC in this country, to make, maybe never late, to make a concluding remark. Thank you.
Thank you very much, Mr. Akile, the MC. Colleagues, also I want to apologize for coming late. I was in another meeting and I thought I would have come here much earlier. But I'm glad I'm here. Colleagues who are here, called one of my senior colleagues, Dr. Ngogo. Thank you for being here. And of course, Andrew, I'm sure that you have given your best when you are talking about seeds, genetically modified organisms. Andrew and many other scientists, our people who do this work at the laboratory level. And my plea has always been to this nation for us to believe our scientists and give them the opportunity to do this, but with a keen eye as to whether they are doing the right thing. I've been at the National Biosafety Committee since its inception, 1996. And I've been chairman now for the last 9 years. Probably I'm overstaying, but I'm happy to say that what I might have missed from what Andrew said, when we are talking about clean seed, there's clean seed at the biotechnology level, which can be at the laboratory level, the tissue culture level, where you produce a seed which is clean, which is readily available in sufficient quantities, and without suffering the vagaries of bad weather. It's also important that this clean seed has been tested for many other things against to make it a productive seed. And I'm sure that this was mentioned. It's also important that this seed is sustainable. In other words, can it be available all the time? We know that anything that you utilize biological system, once used for a long time, it tends to change. Recessiveness tends to come. The negative attributes begin to appear. So you need to redo it and ensure that the good things are brought up again. And that's why in our biological lives as human beings, you don't marry your sister, you don't marry your mother, you don't do those things. Because the negative things will come. So you need to go out and look for good genes. That's why biotechnology to me is not a new thing. Human beings do it all the time, they're looking for it. If you want a nice face, marry a beautiful woman. If you want a tall kid, marry a tall woman. Although occasionally maybe at some point you might get a short one. Because the genetic code might choose a short one out of a very tall person. And that's why when you have a seed, you don't get surprised if you get the opposite. Because you thought you had selected for the best. But unfortunately, the gene on the other side selected the lowest side. Possibly shortness, not resistant to disease, and all these things. In genetic science, that's called take, has the gene taken as its own, and therefore can it do its work. And that's why this has got to continue being done over and over again. The seed obviously must be productive. A productive seed means it yields what you're expecting. And you know the best assessor of yield is the farmer. The farmer knows what they had yesterday and they know what they expect today. And if it's slightly better than that, they will definitely go for it. It's also important that that particular seed should be productive in so many ways. The ease of using it or production. You should not weed it for so many times before you can get a product. And that's why biotechnology helps in these production mechanisms, the weeding. If it matures quickly, it's able to fruit quickly, then you reduce labor, you reduce weeding. And these are all productive indicators. And any seed which gives you that opportunity is a good seed. It of course should be robust to disease, robust to the environmental conditions, drought, salt, pests, and so on. And of course, lastly, if you have used these productive seeds for a long time, you begin to get to know them, you begin to domesticate them. That's why our farmers have this information. The land races Dr. Kimundu was talking about. If you look at cassava land races that have been domesticated, people know them, they like them, they want them to be there. And if you bring another one which they don't like, you have a problem. You've probably known that when distributing cassava stems, some cassava stems have dried up in some Gomoro or somewhere. Nobody goes for them. Because they know that it is not what they want. If they have tried it, there is disease, it's not robust, it does not yield well, and so on. So my hope is that in this discussion which we had, you not necessarily learned anything new, you just reminded it yourself, because this is continuous education. We need to remind ourselves of this and to correct What you thought you knew yesterday, maybe it's not the same today. And so when you come and listen to our colleagues, Dr. Kigondo and other people, it is nice. I only want to make an observation that this forum has obviously declined in attendance, you can see, but there's also one thing which is very clear. The age group of the attendees is getting to be younger, which is good. First of all, the fact that they're interested and they're here, that's good. But also the other reality is that the older people who used to attend these things are no more. And that's why I like to mention with a sense of sorrow the personal way of my colleague, Professor Kermire. Professor Kermire used to come and sit here. He used to be a member of the NPC. And I learned on yesterday that he had passed on while I was in India. And for me, I've known him since we were in Tessa College as far back as 1966, 65. We were there together. And very sad. So when you come here, if you can see your colleague, shake their hands and feel good. Because we are not permanent in this world, we all come and go. Those are the remarks I have been able to come up immediately. I want to thank you for this forum, and also ask other colleagues to come along with you. Even if they are not scientists, the main thing in this world, the older you get, the gravitate to the farm. You gravitate home. Whether you are a president, whether you are what, the older you get, the more you gravitate to the farm. And all of us are begin that early now. By coming here, you learn these things and you appreciate the destination. I thank you.
Ladies and gentlemen, with that, there is nothing useful to add. Our offer is ended.